Ei, 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 oh, 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 ei, ei, sí, 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 eh.
Ei, ei, ei. Está tudo a menos 12. Ei, som. Tô, 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 tô.
O quê? Vem atrás, atrás do microfone. Atrás, a ligação do cabo com a headset.
Não, peraí. Dá-me lá mute. Ei, ei, ei. Não, pá, isto está tudo bem encaixado. Será RF? Se calhar é RF. Ei, ei. Isto vai estar aqui muita gente a ver? Vai estar muita gente aqui na plateia? 60.
Fala, Juan.
Som, som. 1, 2. Ei, 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 ei. Teste, teste, teste. Som. 1, 2, 3. 1, 2, 3. Som, som. Ei, ei. Ei. Estes bacanas vão estar aqui são portugueses ou são estrangeiros? Som, som, som. 1, 2, som. 1, 2, 3, 4. Ei, 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 ei. Som, teste, som. 1, 2, 3. 1, 2, 3, som. 1, 2, 3, som, som. Ei, teste, 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 som. 1, 2, som. Ok.
Troca para botar headset.
Vou para o 3. Ei, 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 ei. Teste som, teste som. 1, 2, som. Oi. 1, 2, 3, 4. 1, 2, 3, 4, 5, 6. Teste, teste, teste. Som, som, som. Ei, ei. Ei, ei, ei. Som, 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 som. 1, 2, som. Teste, teste, teste. Ei, 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 ei.
Consegues ouvir o Frontfield?
1, 2, 1, 2, 1, 2. Som, som, som. Consigo.
Anda à frente, a ver.
Som, 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 som.
Anda em frente do Frontfield.
Som, 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 som. Ei, 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 ei. Sim, ouve-se bem. Sim, sim, sim. Som, som.
Não ves diferença com a coluna de acima?
Som, 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 som.
Aí, vou
Teste, teste,
Fala e vou tirar. Fala.
teste, som, som, som. 1, 2, som. Ei, 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 ei. Som, som. Um, dois, som. Um, dois, três, quatro. Um, dois, três, quatro, cinco. Som. Teste, teste, teste. Som, som, som. Ei, ei, ei.
Rubén, está desligado o amplifier.
O quê?
Atrás, atrás na regi, tens dois amplificadores. Tem que ligar o primeiro da cima, que é o PA.
Pá, eu acho que tem luz. Ei. Ei. É só o amplificador de cima, não é? Acho que já está. Som, som, som. Teste, teste, teste. Som, som, som. Teste, teste. Ei, ei, ei. Som, som, som. Teste. Um, dois, três. Um, dois, três, som. Ei, ei, ei. Teste, som, teste, som. Um, dois, três, quatro. Um, dois, três, quatro, cinco. Ei, ei, ei. Som, som, som. Um, dois, três. Oi. Queres falar quatro?
Espera aí, quero experimentar o PA.
Ok. Ei, ei. Som. Um, dois, som. Um, dois, som, som, som. Teste, teste, teste. Ei, 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 ei. Teste, som, teste, som. Um, dois, som. Um, dois, três, quatro. Teste, som. Um, dois, som, som, som. Ei, 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 ei. Teste, teste, teste. Ó Duarte, não havia nada que era para montar ali no wall? Já dá? Som, som, som. Som. Teste, som, teste, som. Ei, ei, ei. Teste, teste, teste. Um, dois, três. Um, dois, três, som.
Fiz, fiz. Troca para outra.
Um, dois, som. Teste, teste, teste. Ei, 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 ei. Teste, som. Dois, som. Um, dois. Teste, 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 teste. Ei. Dois, três, quatro. Dois, três, quatro, cinco. Som. Dois, três, quatro, cinco, seis. Dois, som. Teste, teste, teste. Ei. Som, som. Dois, três, quatro, cinco. Dois, três, quatro, cinco, seis, sete. Som, som, som. Teste, 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 teste. Som, som, som. Ei, 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 ei. Som, som, som. Dois, três, quatro. Dois, três, quatro, cinco. Dois, três, som.
Boa, troca para outra.
Dois, som. 1, 2, 3, 4. 1, 2, 3, 4, 5. Som. Teste de som, teste de som. Ei, ei, teste de som. 1, 2, 3. 1, 2, 3, 4. Som, som. 1, 2, 3, 4, 5, 6, 7. Som, som, som. Teste de som, teste de som. 1, 2, 1, 2, 1,
Anda,
2.
Rubén, anda ao lado do púlpito, por favor.
Teste, 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 som, som. 1, 2, som. 1, 2, 3, 4. 1, 2, 3, 4, 5, 6, 7. Som, som. 1, 2, 1, 2, 1, 2. Som, teste, som, teste, som. 1, 2, 1, 2, som. Queres testar o 6? O backup? Som, teste, som. Ei, 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 ei. Teste, 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 som, som, som. Som, teste, som, teste, som. Teste, 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 som, som, som. 1, 2, 3, 1, 2, 3, 4. 1, 2, 3, 4, 5, 6, 7. Ei.
Hermoso, hermoso,
Diz?
hermoso, hermoso. Bonito.
Estou bonito.
Não, tu não.
Olha, queres testar os de mão?
Por favor. Ontem testamos os demás hasta que acima y daban. Ontem experimentamos os microfones de mão até aqui acima e davam.
Ok. Eu vou andar. Um, dois, som. 1, 2, 3, som, som. 1, 2, som. 1, 2. Olha, eu ponho. Veja o que é que acontece. Som, som, som. 1, 2, som. Teste, teste, teste. Som. 1, 2, 3, 4. 1, 2, 3, 4, 5, 6. Som. Som, som. Teste, teste, teste. Som. 1, 2, 3. 1, 2, 3, som. Ei, 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 ei. Som, som, som. 1, 2, 1, 2. 1, 2, som. 1, 2, som, som, som. 1, 2, som. Ei, teste, teste, teste. Som, som, som. 1, 2, 3, 4. 1, 2, 3, 4, 5, 6, 7. Teste, teste, teste. Som. 1, 2, 3. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, 6. Som. Teste, som. Ei, ei, ei. Som, 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 som. 1, 2, som. Teste, teste. Som, som, som. 1, 2, 1, 2, 1, 2. Som, som. Teste, teste, teste. Ei. Teste som, 1, 2 som, teste, teste, som, som. Ei, ei, ei. Teste, 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 som, som. 1, 2, 1, 2, 1, 2, 1, 2, som. 1, 2, 3, 4, 1, 2, 3, 4, 5. Ei, ei, ei. Teste, 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 som, som, som. 1, 2, som. 1, 2, 1, 2, 1, 2, teste, 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 teste. Ei. 1, 2, 3, som, som. 1, 2, 3, 4, 5.
Tá, tá.
Agora o azul. Ei, ei, ei. Som, 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 som. 1, 2, 3. 1, 2, 3, som. 1, 2, 3, 4, 5. Som, som, som. 1, 2, 3, 4, 5, 6. 1, 2,
Tá.
3.
Tá. E, 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 O, 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 E, 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 E,
3, 4, 5. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5.
Good morning, everyone. We would very much like to welcome you all to the auditorium of the Champalimaud Foundation. Minutes ago, you were welcomed by our president and vice president, Leonor Beleza and João Silveira Botelho, at the Champalimaud Center for the Unknown, at the entrance of a building which harbors both the Champalimaud Clinical Center and Champalimaud Research. My name is André Valente. I work with the board of the Champalimaud Foundation, on behalf of which I will be guiding you through both what we do here and a discussion and series of presentations of my colleagues on some of the most fascinating discoveries and greatest challenges that lie ahead of us in society as much as in science. We will have 4 short talks by our leading researchers, after which we will sit and open the room to questions and discussions from all of you. So keep your questions and your issues to the end, and we will be able to discuss them. I will start right away by introducing our first speaker. Our first speaker today and co-director of Champalimaud Research is Joe Patton, and I will be introducing each one of the speakers as they come and speak. Joe received his undergraduate degree in biology from Tufts University in Boston, and his PhD in neurobiology and behavior from Columbia University in New York. He performed his doctoral work in the laboratory of Daniel Salzman, where he studied the neural mechanisms that underlie learning about positive and negative value in the primate brain in what we call the limbic system. In 2008, Joe initially joined Champalimaud Neuroscience program as a fellow and became a principal investigator in 2012. Here at Champalimaud, his lab seeks to understand the algorithms and circuit mechanisms underlying cognitive behaviors with a particular focus on temporal processing, so how we perceive time and process it in our brain, and reinforcement learning. He is a Howard Hughes Medical Institute International Research Scholar. He's an alumnus of the Simons Foundation collaboration on the global brain, and he's also currently a member of the executive board of the Fulbright Commission in Portugal, and a holder of an European Research Council Consolidator grant, a grant which I am sure many of you are familiar with. At Champalimaud, Joe has been the director of the International Neuroscience Doctoral Program since 2017, as well as co-director of the Cajal Advanced Summer Training Course in Computational Neuroscience. He became the director of the Champalimaud Neuroscience Program in 2019. Thank you, Joe. Please. You can take your mask off.
Thank you, André. As André said, my name is Joe Patton, and I represent the neuroscience arm of Champalimaud Research here at the Champalimaud Foundation. What I want to do today is just briefly give you a sense of who we are, what we do, why we do it, and a little bit about how we plan to leverage our successes so far towards furthering the ultimate goals of the Foundation to improve life by tackling some of the hardest problems we face in science and health today. Now, of course, like the Foundation itself, our program is relatively recent, having only been founded in earnest in 2008. And since the beginning, our central focus has been on fundamental research into the mechanisms of behavior. So what is fundamental, sometimes called basic or pure research? And why do we invest in it? This is research for the sake of understanding, for creating knowledge about how the world works. It's looking up at the night sky and wondering why the stars move the way they do, not because you want to use that information to navigate around the world, but nonetheless, absolutely necessary for developing the ability to do so. Once able to navigate, of course, new discoveries can be made. This positive feedback relationship between fundamental and applied science is the engine that drives the development of technology, tools, and in the case of biology, treatments for disease. Even the name of this center we're sitting in, the Champalimaud Center for the Unknown, pays homage to the critical importance of fundamental knowledge, understanding, and discovery in charting our course forward towards a better future. Now, the penumbra surrounding the pure neuroscience research we do here is a space of enormous potential, and we hope to capitalize on this potential more and more as we move forward. mindful that this depends on protecting curiosity-driven discovery science. Now, within the neuroscience program, we look not to the stars for understanding, but inward at behavior and the mechanisms that produce it. And that is because it's behavior that created this marvelous organ, the human brain. Embedded in its circuitry and chemistry is you and all of us. It is the human condition, the source of our experience, our thoughts, our personalities, and our social interactions. And the reason we have everything we see around us today that is the product of human ingenuity, science, engineering, culture, medicine, all because of this organ. And yet, we're still so far from unlocking its secrets, secrets that not only might help us find better treatments for a range of diseases, but to build a deeper understanding of natural intelligence that can be used to create more robust and flexible artificial intelligence that promises to transform the human endeavor forever. So how do we unlock the secrets of this marvelous and complex organ? As I mentioned, our approach starts with behavior, because behavior, broadly defined, is why the brain evolved at all in the first place. It's also a lever we use to interrogate its function. I want to unpack that a little bit now. Behavior drives intelligence. The roboticist Rodney Brooks, formerly of MIT's AI lab, made this point in a now classic paper titled "Elephants Don't Play Chess." In this paper, he argues that the failure of AI to produce really general, robust intelligence, and believe me, despite all the promise of current AI methods, it's still a failure, is because we weren't focusing on the hard problems that nervous systems face during evolution. That by jumping to problems like solving strategy games, we're ignoring natural history. That's a history where the first life forms appeared on this planet approximately 4 billion years ago. The first nervous systems, 600 million years ago. It then took 599,750,000 more years, give or take, until just 250,000 years ago for modern humans to appear. So, the human brain, this pinnacle of evolutionary ingenuity, has only been around for 0.04% of the time that nervous systems have existed on this planet. He argues that problem-solving behavior, language, expert knowledge and application, and reason are all rather simple once the essence of being and reacting are available. That essence is, for example, the ability to move around in a dynamic environment, which we can gain more mechanistic insight into by studying organisms other than ourselves. Here, you can see 2 examples of how researchers here are characterizing locomotion in mice and flies, applying sophisticated machine learning algorithms to rigorously trap positions on the body to characterize the output of the nervous system as animals move around in their environment. This kind of rigorous and detailed quantification of behavior will be critical for ultimately determining how neural circuits contribute to motor control and coordination. This essence also includes being able to sense and perceive the surroundings to a degree sufficient to achieve necessary maintenance of nutrition and hydration, such as this mouse here that localizes this drop of water using its nose and whiskers, initiates and guides a reach, and grabs the water and brings it to its mouth. These kinds of behaviors can seem mundane, but this part of intelligence is where evolution concentrated its time. It's much harder, and once mastered, it formed the building blocks for abstract intelligence, symbolic reasoning, and yes, the ability to invent and play the game of chess that we see in humans. To understand the computations the brain performs to accomplish such tasks, we can use tools that, when you step back from it, really seem like science fiction, to literally see what the brain is doing during the execution of these behaviors. In this video here, you can see cells light up when they're electrically active due to the expression of a genetically encoded activity indicator. So these kinds of experiments produce incredibly rich data sets with behavior on one side and the activity of up to tens of thousands of neurons on the other that we can use to determine the neural basis of a variety of functions. Neuroscientists here developed cutting-edge virtual reality paradigms that allow them to take experimental control over the environment and measure the impact on animal behavior. Here you can see one such example of a fly whose position is tracked as it moves around an arena, and that position is used to update the visual environment projected into that arena. The way the animal's position and heading are mapped to the projection can be modified and the resulting behavior observed to understand how visual feedback modifies motor control. Groups study a range of problems here, ranging from sensory motor coordination, as I've described, to decision making, such as this example here, where the food choice behavior of flies with different internal nutritional states can be observed and quantified. You can see on the left here is a fly that's fully fed, on the right, one that's been deprived of protein. The fly on the right spends the majority of its time eating from food sources that are spotted around the arena. How internal state modifies neural circuit function to guide adaptive behavior is an incredibly important question that, if understood, could help us combat everything from obesity to addiction and depression. Other groups study problems related to social behavior, such as in this video on the right, where the schooling behavior of zebrafish is studied to try to understand what are the behavioral rules that the brain of each fish carries around to ultimately create the emergent dynamic behavior of the group. And here comes an opportunity to highlight an example of the synergies between fundamental and applied science. In this video here, you can see that each fish is labeled with a number. That labeling was performed by a machine learning algorithm that was developed here at the Foundation. It turns out these kinds of multi-agent tracking problems are hard ones because collisions between individuals can often confuse the algorithms. After building this model, a schematic of which you can see here, the group began to develop a new theory of deep networks based on abstract algebra to replace the optimization schemes that are the current state of the art. The potential applications are extremely broad. As part of a European consortium, this theory is being implemented in hardware and used to characterize human and robot behavior. This is just one example of how basic questions can lead to technological breakthroughs, proof that applications are not necessarily arrived at through seeking them out in the first place. Recently, the Champalimaud Foundation was ranked by Nature Index at number 4 in the world amongst nonprofit institutions in the area of artificial intelligence, in large part because of the way neuroscience researchers here are applying AI algorithms as tools to gain insight into data, but also because of the deeper conceptual connection between the computational models of brain function that we develop to understand behavior and AI. This last function is supported by a number of theoretical and computational neuroscience groups within the neuroscience program. Here you can see, in this video on the left, activity from almost the entire brain of a fish being presented with visual stimuli, moving in the direction indicated by the arrow. Color here represents depth into the screen, and brightness reflects fluorescence from a genetically encoded calcium indicator. Now, calcium is normally very low in concentration within neurons. When they fire action potentials, however, these all or none electrical impulses that are the unit of long-range information transmission in the brain, calcium levels rise rapidly, and this can be literally seen through the light emitted by the indicator. Because the fish's brain is transparent, unlike most other brains, researchers can watch the whole global brain network at work to try and understand how it's processing the visual stimuli or performing other functions, depending on the experiment. On the right is a similar large volume calcium imaging experiment, this time in the brain of a fly, that's sampling different foods. By experimentally manipulating the fly's internal state, researchers are uncovering the manner in which sensory and motor circuits are modified by internal variables to alter the food choices that animals make. Not only can we use these kinds of experiments to understand how vision works or why food tastes better when you're hungry, but we might be able to build better robots to navigate using vision or find ways to help people make better dietary decisions. Now, we would be here all day if I were to outline all the discoveries and developments here over the last decade or so, but just to give you a bird's eye view, we in the neuroscience program leverage deep interrogation of behavior together with the development of new methods for observing and measuring the activity of the nervous system, as well as theory to help us develop principled and compact descriptions of the data. Ultimately, this is to try to define the neural mechanisms underlying things like perception, decision-making, cognition, and movement. And I purposely not singled out individual research labs in the presentation, instead opting to present our approach as a collective one. We are currently 12 group leaders, many of whom you saw the work of, at least briefly today. But what we do here goes beyond the knowledge we produce or even the applications that ultimately might be derived from the understanding we generate. I mentioned the word penumbra before. Science is a way of understanding the world. And perhaps I'm biased, but I think it's the best we've got. And so I think we have a responsibility to help spread our processes, our methods, our ways of thinking beyond the laboratory. And we try and do this in a number of ways. One is through education. We've established an innovative and widely recognized graduate program in neuroscience, and we are expanding this to include other programs. It gives students both classroom, curriculum, and project-based practical learning, as well as deferred selection of doctoral thesis labs. This gives students the time and space to develop into deep, capable scientists. And these are skills that go beyond the laboratory. Society in general needs rigorous, evidence-based decision-making. Science is a great training for that. We also organize and host the premier advanced summer training courses in neuroscience, the Fens Cahal Summer Schools, which draw students and instructors from all over the world to spend an intense few weeks together learning about the latest in a variety of subfields. We've developed our own brand of robust science outreach. This includes our series of AR events, Portuguese for air, that seeks to bring science and society closer together by examining issues of popular interest. And lastly, there's a lot of capacity to develop around the process of doing research. At the outset, it was decided that functions and expertise likely to be useful widely in the community should be concentrated in shared core facilities and units. In that vein, we've established a set of scientific and technological platforms, as well as administrative support units. The services originally established to support research are more and more being sought out by all corners of the foundation, and that's something we'd like to further enhance to help broaden the impact of the work we do. So just to wrap up, I've told you a bit about our goals, our motives, and activities from the perspective of the neuroscience program. I'm very excited by what we've been able to accomplish both within our ranks and together with others throughout the foundation. So is the European Commission, that's awarded us with 12 ERC grants so far. But ultimately, we're just one entity in a larger ecosystem that is the Champalimaud Foundation. As we move forward, we look to build even more bridges between neuroscience research and the other areas here, to listen to clinicians, researchers, and administrators around us about the problems they seek to solve, to try and find synergies where they might not have been appreciated earlier, through shared interest in technology, through the science, or perhaps by appreciating that the fact that things like diet, sleep, social interaction, exercise, all fundamentally reflections of behavior, seem like some of the most broadly important factors affecting human health, including cancer. Perhaps my colleague Enrique can tell you more about that in a few moments. Hopefully by the time you visit us next, I'll be able to tell you about all sorts of new bridges we've built between ourselves and into the unknown. Thank you.
Thank you so much, Joe. Thank you for this wonderful exploration of the brain and what we do here. I will start straight away inviting our next speaker. And Henrique Veiga Fernandes, I'm very happy to introduce him as the director of our Physiology and Cancer program. Henrique graduated in veterinary medicine at the University of Lisbon. He was awarded a PhD in molecular and cellular biology from Université René Descartes in Paris, France. And he developed his postdoctoral research at Institut Necker, also in Paris, and at the National Institute for Medical Research in London, in the UK. He first started his own group in 2009 at the Institute of Molecular Medicine in Lisbon, and was a member of the IMM board of directors since from 2014 to 2016. He joined us at the Champalimaud Center of the Unknown as a senior group leader in 2016 and became co-director of the Champalimaud research in 2019. Enrique has made pioneering discoveries in immunological memory and neuroimmune interactions. Among multiple distinctions, he received several European Research Council awards. He has been elected member of the European Molecular Biology Organization, EMBO, in 2015, and he was elected an Allen Distinguished Investigator in 2018. Already this year, in 2021, Enrique was nominated to the scientific board of the prestigious Science magazine. Thank you so much, Enrique, for joining us.
Thank you, Andre. I'm just waiting for the presentation. Here it goes. So around the 1700s, in a very small city in the north of Italy, something happened that really brought to the world a new sound. That small city, probably some of you know the city I'm referring to, was Cremona. And the truly technology revolution took place in this city from the 1600 to the 1700. It was a technology revolution in music and on instrument making. And the likes of Antonio Stradivari really brought to the world, in the time of a generation, probably the most desirable musical instruments that we have to date, the famous Stradivarius violins. This was really perfection in technology manufacturing. This was a tremendous opportunity at that time for composers, but also a challenge, an opportunity because there was a new sound that composers could use to create wonderful music for the generations, for the years to come, and centuries. And it comes without a saying that composers such as Vivaldi or Bach are still major references of the Baroque music nowadays. But it was also a challenge, and a challenge because technology by itself cannot make a revolution. You need the genius, the human genius, and creativity that is so inherent to human talent. My name is Enrique Vega Fernandez, as Andrés said. I'm a senior group leader at Champalimaud Foundation, and I'm also the director of the Champalimaud Physiology and Cancer Research Program. And what I would like to take you today is into a small trip into what we're doing in cancer research, and by demonstrating or giving you examples that by investing in transformative and advanced technology and in recruiting and attracting world-class talent, we can make a difference in cancer research, cancer care, and cancer treatment. Essentially, our physiology and cancer research program, it's a fundamental or basic research program, similar to what Joe just mentioned about the neuroscience program, where we're really focused on providing transformative, basic research knowledge into the understanding of cancer. This is a tremendous opportunity in a setting such as this one. World-class platforms and technology facilities, a wonderful clinical setting where we can discuss and get information from our clinicians. But it's also a major challenge because different institutions such as this one are tackling the problem of cancers throughout the world, in Europe, in the US, in Australia, everywhere. So we really needed to find a way that would be different, a different angle to tackle that problem. And what we've been doing really in the physiology and cancer program is trying to bridge the gap between our basic high-impact science into different clinical applications. So cancer is actually a very complex disease, and cancer itself is an extremely heterogeneous disease, and you will hear more about that, certainly from Rita and from Marcus. You can see from this picture that the different colors you're getting are actually within one primary tumor, different cell types that compose the tumor, but also represent different biological activities of that tumor. So whenever we think about one specific type of tumor, we're actually dealing with multiple and very different cell types with distinct genetic signatures and interacting with a certain number of other cell types that make this disease so difficult to tackle and to cure. And this is really related, this heterogeneity, with a certain number of features or hallmarks of cancer that actually make this disease so complex. Cancer development actually does not occur from one day to the other. Frequently, it's a silent disease that takes decades to happen. This all starts with initial mutations that occur in a few cells in the body, and frequently, in a silent manner, over decades, there's cancer progression. These cells, these so-called stem cells, cancer stem cells, they divide and reach a certain point where the size and the features of this cancer are such that the tumor can metastasize and provide metastases that then disseminate cancer and this disease to the whole body. So it's a disease that normally starts in very discrete tissues or organs, but then over the years can become a systemic disease. So in the cancer research program, what we're doing is actually taking advantage of very advanced technology experimental models to interrogate how tumor cells develop, how do they interact with their environment, and what are the strategies that we can implement to eventually find new avenues for cancer therapy. To that end, we're using experimental models that actually range from very simple organisms, such as the fly or the fruit fly, down to mice models or mammal models, where we can actually study the behavior of cancer cells and even human cells. You will hear from Rita that fish can also be used to study the behavior of human cancer cells. But the take-home message from this slide is actually that cancer is a very complex disease. And to understand how a cancer cell or cancer itself behaves in an organism, we need to understand the complex tumor environment. A tumor cell does not resist or does not evolve in the human body isolated. It actually evolves in these complex type of scenarios depicted here in this beautiful painting that you can visit in Lisbon in the Museu de Arte Antiga. Actually, a cancer cell needs to interact not only with their peers, with other cancer cells, but it also needs to interact with other systems in the body, such as the immune system that is so important to fight cancer and to fight other type of infectious disease. So understanding how cancer cells interact with other cells in the body and understanding how other systems in the body can control cancer development is actually a very important topic in cancer research. And I want to give you two simple examples of how, from basic high-impact fundamental findings in our program, we actually can make a quite fast bridge into the development of new classes of drugs for cancer treatment. The first example I would like to give you comes from my friend and colleague, Eduardo Moreno. Eduardo, over the last few years, about 5 years or so, made groundbreaking contributions on the understanding of a process that is called cell competition. And in cell competition, and to define and describe these tenets, he took advantage of a very simple mouse model, the fruit fly, that likely some of you at some point in time already had in your households. And what they discovered was that mouse cells can express molecules or proteins on their surface that allow fly cells to communicate between each other. And frequently this comes in two flavors. A flavor that is called UB. This is a flavor that gives a competitive advantage of this cell on the left-hand side. And then another flavor that is called loose. And whenever these cells interact, the loose cell dies while the UB cell survives. So it comes to no surprise that this is a conserved mechanism throughout evolution and found in different species that is so important not only for the development of multicellular organisms, but also in aging. So this was actually two major discoveries published in 2015, but very rapidly the group of Eduardo actually tried to bridge this fundamental discovery and put it in practice in human cells. And what you're seeing in this video that has been playing and looping for a while is actually an example of human cells in a petri dish, where you can now make cells expressing this UV flavor, and these will be the red cells, while the cells expressing the loose flavor will be the green ones. And you can appreciate that over time of this culture that will start now, the winning cells, red cells, will become dominant while the green cells will start disappearing, indicating that cell competition using these principles can be harnessed for potential therapy. And in what type of diseases? Cancer. So 2 years ago, Eduardo published a transformative paper for the understanding of cancer, where they actually harnessed the potential of these very basic and fundamental biological process in cancer therapy. So they basically block the use of what I would say an anti-competition therapy in tumors. You can appreciate that when they treat animals and different types of tumors with this therapy, the tumors that develop are significantly smaller than the placebo, and with an effect that is fairly similar to chemotherapy, standard chemotherapy that is used in the clinics. But if now they combined standard chemotherapy with this novel class of therapeutic approach, now the effect is quite dramatic in tumor development, demonstrated that in a very short period of time, less than 1 decade, we can go from very fundamental discoveries in simple experimental models to a full package of preclinical data that can instruct the development of new classes of drugs. The second example I wanted to give you actually relates to research that comes from our own lab, where we've identified over the years with a certain number of seminal papers that in peripheral tissues, such as the intestine that is depicted here, but we can find it in multiple other tissues, such as the lung, the pancreas, or adipose tissue, there are 2 systems that actually are very important for the proper development and function of these organs. One of them is the nervous system, and you've heard from Joe the importance of the central nervous system in defining behavior, but the central nervous system is just one portion of the nervous system, and we find it everywhere, not only coordinating behavior or motor behavior, but coordinating multiple other functions. The second important system was the immune system, and you can see here the nervous system in red, the immune system, or part of some immune cells, in green. And you can appreciate that they are closely located in developing organs, suggesting there's actually a potential interaction between these cells. And more recent research, actually from the lab, actually demonstrated that not only they are in close proximity, but they have a very intimate interaction. They establish these very close interactions between neurons in the intestine, for instance, or other organs, and the immune cells that allow them to crosstalk and influence the function of each other. And if you think about how neurons can impact on immune cell function, then the bridge actually to start and developing new classes of drugs is obvious. So one of the very important unmet needs in treatment of cancer and in minimizing risk of cancer development is to tackle chronic inflammation, a major trigger of cancer. So we actually started by triggering or harnessing what I now call neuroimmune therapy in the context of chronic inflammation of different organs. And I give you just 2 examples. One of them is lung inflammation on the left-hand side. So if you treat animals with placebo, nothing really happens. You have a very inflamed lung with this red color and a lot of tissue destruction due to inflammation. But if you treat it with this neuroimmune therapy, you basically go back to normality. You establish the right balance of that organ. The same thing happens with intestinal inflammation. On the left-hand side with the placebo, you have a disrupted, completely altered morphology of the intestine. But if you harness these neuroimmune interactions, now you can cure animals and you get back to an absolutely normal structure of the intestine. So as I said, chronic inflammation is probably one of the most worrying risk factors for the development of cancer, and it tightly associates with what now are called the civilization diseases, such as obesity and metabolic disorders that induce a chronic state of inflammation. So it comes without a saying that these neuroimmune interactions, or we thought in the lab, could potentially be used for harnessing in the treatment of cancer. And this is indeed what I wanted to show you, that in the context now of a liver cancer, if we treat animals that develop liver cancer with this neuroimmune therapy, as you can see here on the right-hand side, you get tumors that are significantly smaller than tumors that are developing in the absence of treatment or being treated by a placebo. So I'd like just to finish, hoping to have convinced you that a fundamental and disruptive cancer research program can actually make fundamental contributions to a clinical practice. I think we are tackling these and we are achieving these based on three main pillars. The first one is really an investment in transformative and advanced technology that was made at the foundation. The second one is by attracting and retaining world-class talent. We've got amazing scientists that you will hear today. And the third one, and very important, I call it conversions. And this really has to do with the architecture of this building, where we can, on a daily basis, interact with clinicians, hear their interrogations, and learn from them, and ask and use that information to push forward our basic research in an atmosphere or an environment that I would call a fusion research institute. Thank you for your attention.
Thank you very much, Enrique. After having heard from two directors of Champalimaud Research, our next speaker is Rita Fior. Rita is a developmental biologist, and she's fascinated with how cells interact. How cells interact with each other to deliver instructions to each other, to sometimes collaborate, other times compete, to synchronize, and to work together in a way that builds our organism. Rita is a developmental biologist, so she first started learning how an organism is born from a single cell and what the forces that do this are. During her PhD, she studied not downstream signaling events, that's the scientific name. They are events that control the balance of neural differentiation, how the nervous system differentiates, and how the mother cells, these progenitors, are maintained and generated in what is called an unstable notch reporter that she developed to study these mechanisms in real time. So she observed the processes as they happen. For a postdoc, she started using zebrafish as a model organism, and Enrique just mentioned how the fish can actually be used, and Rita will tell us about it. And during this work, she found a specific feedback loop that has to do with RNA, and that coordinates three major events during segmentation, which is when an embryo starts being divided into distinct parts and first acquires these segments. There's differentiation, cell movement, and stem cell maintenance. These are all vital processes. More recently, she became interested in cancer. And this turn in her scientific interest started building up with her own disappointment on how patients and clinicians often face chemotherapy choices for advanced disease with little information to decide what may work best in an individual. I will not tell you about it. She will. I can just say that ever since she first published the zebrafish xenograft model, and she will explain it, for personalized medicine in 2017, she has been rapidly advancing. With her findings widely cited and the image she captures for her work, which are both beautiful and eerie, are featured in magazines such as National Geographic. With Rita, you will be introduced to an individual scientist's quest to conquer disease and find better and quicker pathways to effective life-saving therapies. Thank you. Thank you very much, Rita, for joining us today.
Thank you. Thank you, André. I don't know if we need to put down the lights a bit more, but thank you so much for the opportunity to talk about the work we've been developing in the foundation here. So my lab has two main missions. One is really to develop a personalized test for anti-cancer treatment that I'm going to talk about using the zebrafish model. But also, we have another mission which started with curiosity-driven research, which is really to understand the interactions between the tumor cells and the microenvironment, and actually how cancer cells are able to avoid or evade the innate immune system, which is the first responders. But I'm mostly going to talk about the first one. So I always start with this slide just to make a point, which I think is already made of it, which is the fact that we are surrounded by amazing technology. We can fly to the moon, we can fly to Mars. I'm going to take out this, sorry. Hello? Oh, I bring this, sorry. Hello?
Keep speaking.
Sorry? Hello. Sorry for this. So I was saying that we are surrounded by amazing technology. We can fly planes every day, we can fly to the moon, we can sequence genomes every day, we can make a vaccine in 1 year, which I think is absolutely amazing. But if you're a cancer patient, there is no test to tell you whether the treatment you're getting will actually kill the cells, kill your cells. Because many times doctors have options, equivalent options in their hands, A and B, but there is no test to actually test your own cells whether A or B is the best. And this happens with bacterial infections. You can do a test like an antibiogram to tell you which is the best antibiotics, but this is not happening to cancer treatment. And this was really my main driving force to start this project, to actually develop the zebrafish avatar project. So when I started this project, actually, the gold standard in the field to actually test each patient's tumor was to use the mouse model, which we already heard about, which is a great model where you take tumor cells from patient, you put it in the mouse, and you have an avatar. And then these mice can be tested, which is the best treatment for that matching patient. The problem of the mouse model is that it takes months to develop and to have an answer, and patients don't have months to wait. So that's why I started developing the zebrafish avatar model, where you can take tumor samples from patients, either from a surgery or from a biopsy. You label them fluorescently, and then we can microinject little zebrafish embryos of 2 days post-fertilization. And then after 1 day, we can start treatment with chemotherapies or even radiotherapy. We developed a protocol to take the zebrafish to exactly the same machine as the patients, or biological therapies. And the idea is that after 3 days, we can look at these tumors from the patients and see which is the best treatment for that particular tumor. We want to see if the treatment is inducing cell death, so killing the tumor, reducing the tumor size, see if it can block angiogenesis, which is the process of recruiting new blood vessels into the tumor, or if it can block micrometastasis formation. And the idea is to do this around 2-3 weeks, we would have a result. This is our dream. Our dream is to have these tests and help MDs choose the best option for their patient one day with a test. This would avoid patients going through round of trial and error approaches, because usually what happens is if you don't get the right treatment at the first time, then you go to the second round of treatments and third and so on until you find the best treatment. And this is exactly what we're trying to avoid. So this project that we are developing here at Champalimaud Foundation is only really possible because we are collaborating with all these people. So we, the scientists in the lab, are collaborating with the pathologists, the immunologists to take the biopsies, or the surgeons, radiologists, physicists, oncologists, the nurse. So it's really a multidisciplinary endeavor, which is really possible in this building, but also we also have collaborations with other hospitals here in Lisbon. So we, until now, have been developing protocols for colorectal cancer, pancreatic cancer, stomach, breast cancer, ovarian, bladder cancer, but also for targeted therapies. And so we'll give you a bit of the roadmap and how we do this, how we do the assay, why the zebrafish, and show you a bit of data. I hope it won't be too much. So this is the zebrafish embryo. So this is a little larvae. You can see the eyes. You can see in green the blood vessels of the fish. And this is a tumor that we injected just near the heart. And this is a fin of the zebrafish. The great advantage of the fish, as Joe already pointed out, is it's transparent. So we can look through it. And we can take the whole animal into a microscope. And then we can zoom in into the tumor and actually have single-cell resolution and look at the cells and what's happening to these cells when we put the drug. Are these cells dividing? Are these cells dying? What's happening? And that's really the power of technology, of biology, that we have at this moment in our hands to study biology that we are trying to transfer to the clinic, okay? The other advantage of the fish is that we can actually look at proliferation. This is a marker of proliferation. These cells are actively dividing. We can look at also if cells are able to recruit these blood vessels from the fish, because all the signaling pathways are really conserved, so we can see this. So tumors that are highly angiogenic are really able to recruit blood vessels from the fish. But also, the great potential of the model is that you can actually see whether the tumor can metastasize along the fish. So can migrate along the fish and invade distant organs like the brain. Here you have the eye, the retina, this is the lens, the gills, or the tail of the fish. And this can be a really important clinical data also. Okay, so here I'm just going to give you an example of what I was talking about, the options that doctors can have in their hands. So this is the guidelines for colorectal cancer, therapeutic guidelines from the US, from the NCCN. There's also just the same from ESMO in Europe. And every time you see or, this is an equivalent option. So you can see here all the options that there's in the hands. And only for one of these drugs, there is actually a biomarker. So a test that you can do to actually tell you whether you should have this drug or not. Otherwise, there is no test, okay? But if you look closely, what you see is that if patients start with FOLFOX, it doesn't work, you go to FOLFIRI. If you start with FOLFIRI, it doesn't work, you go to FOLFOX. So the two major arms in colorectal cancer are FOLFOX and FOLFIRI. They're very similar combination of drugs, but they're different. So how do you choose that? There is no actually way to choose that, because in clinical trials, it has been shown that both drugs have the same response rate. Imagine, it's around 40%. 40% for one, 40% for the other. But if you're a patient, you don't know if it's going to go in the 40% that respond, or the 60% that do not respond, or if you take the other way, you'll get the right drug. So we wanted to develop this model. The first thing we need to do is validate and see if it's a good model. It could be a good idea, but it doesn't work. So we really needed to validate the model. And I'm just going to explain a bit what we do in the lab. So we inject and make these little fish with the tumor, which we call the avatars or xenografts. And in this example, I'm just going to show an extreme example, which these two cell lines derived from a patient of colorectal cancer. But the thing is, this was the one that was derived from the patient, and this was developed in the lab by a Japanese group. And the only difference is that these cells don't have one mutation. All the other mutations are the same. So the cells are very, very similar, okay? It's only one gene is different. And as you can appreciate here, so what we do after the treatment of FOLFOX and FOLFIRI is use a marker to look and find the cells that are dying and quantify cell death, whether the drug is able to kill cells or not. So when you see a lot of green, it's because cells are dying. And I hope you can see that HET116 is really sensitive to FOLFIRI, a bit sensitive to FOLFOX, but definitely these cells that are almost sisters do not respond to treatment. And so this was really to try to prove that the fish could be a good model because it has this sensitivity. Now, at this point, what we're doing is really using real data from real patients here from the Champalimaud Foundation. And what we are doing is really comparing the results in the fish avatar and the response of the patient. So we got access to these patients that went to surgery, and then after surgery, what they do is that they have to have a chemotherapy treatment to reduce their chances of having a relapse, right? So what we did, we take the samples from surgery, excess samples that came from the surgery, we label these cells, and then we inject the zebrafish. Then we treat these fish, control, no drug, or with chemotherapy exactly the same as the patient. The idea is really to validate the model, whether this is a good predictive model or not. And this is at the stage that we are. And what we have now, really, with our patients in colorectal cancer, is that around 84% of the cases, we are able to predict the response in the patient, with a positive predictive value of 92% and a negative predictive value of 75%. If we now plot this in a different way, so if we separate the patients whose avatar had a really good response, and we separate from the other patients whose avatar had a bad response, here we separate the two populations. Now it's pretty clear. So the patients that had a good response, their avatar had a good response, have a much better progression-free survival. So these results are really being, I think, very promising results, and we are building on them still, and hope to go for the next phase where we will actually help direct treatment. Okay, I'm just finishing out, just saying that the other arm of the lab and the other mission is really to try to understand the interactions between the tumor cells and the tumor microenvironment, especially innate immunity, which is the first responders of the immune system. I'm just going to briefly explain why I was fascinated by this, because when I injected tumor cells in the fish, and then the other advantage of the zebrafish is that you can put the whole fish live with the tumor in the microscope, and you can look at cells, what they're doing live. So this is a vessel. So in red, you have the tumor cells, and in green, you have the army, the first responders, which are macrophages and neutrophils, the innate immune system of the fish. And I hope you can see that the green cells are trying to chase the red cells, some green cells eating up some red cells, but some red cells are totally invisible. And that's what we saw, was that some human tumor cells, when you implant in the fish, they are there, they're fine, they are not eaten up. But other tumor cells are completely cleared by the fish in amazingly 3 days. Okay? So this is another movie where you can actually see these macrophages eating up all this tumor. And so we are really interested in trying to understand what are the mechanisms that block this, what are the mechanisms that activate this, because in this way we can find new drugs to help immunotherapy, we hope, and take immunotherapy to more patients. So this is what we are trying to do in the lab. Okay, so I'd just like to acknowledge all the team. So I'm pretty lucky to have a really amazing team with me, with students and postdocs and so on. I would like to thank all the collaborators here from the foundation, but also the collaborators from other hospitals in Lisbon, and collaborators, the platforms that already talked here, which is great that we have here at the foundation, really a supportive unit that can help us with our work. And I would also like to thank my mentors, Leonor Saud and the late Maria de Souza, and funding, and you for listening. Thank you.
Thank you very much. Thank you very much, Rita, for a beautiful, beautiful story. During the course of 2018, with the foundation achieving rapid growth and a track record of scientific success, which you've been seeing here, the board, with the generous support of the Boton family, devised a new center. A center that would be dedicated to one of the hardest cancers to treat. One that would benefit from a multidisciplinary approach that Rita's already mentioned, with out-of-the-box scientists and clinicians from many fields. The foundation worked once again with a vast team of scientists and clinicians, and a team of architects and engineers to devise and create the Boton Champalimaud Pancreatic Cancer Center. The project soon became a reality, and the center should be opening its doors still this year. Our final speaker, before we open the room for questions and discussion, will be introducing us to the activity at the center of this new building and some of the challenges he plans to meet. You are seeing now the rendering of the new building, of the Pancreatic Cancer Center, as it was initially devised during the course of 2018. The building, you might have noticed, has been growing. Those of you from Lisbon surely have seen it as you drive by. And today, as you approach the foundation, you can see the new building being born, and it will be inaugurated still this year with more new science and patient care coming into the foundation. I will be introducing, at the center of much of what's happening here, Marcus Maurer. Marcus Maurer received in 1989 his medical degree at the Johannes Gutenberg University of Mainz in Germany, with part of his training taking place at the University of Zurich in Switzerland. He was a research fellow at the Department of Surgical Oncology at the University of Pittsburgh in the USA, and then assistant professor of surgical oncology and immunotherapy at UPMC in Pittsburgh, Pennsylvania. In 1999, he became full professor of microbiology and infectious diseases at the University of Mainz and head of the diagnostic laboratory at the Department of Medical Microbiology. In 2005, he becomes professor of infectious diseases, particularly clinical immunology, at the Karolinska Institute's Microbiology and Tumor Center, also known as MTC. He served both as head of the division of therapeutic immunology at the Karolinska and as senior physician of the Center of Allogeneic Stem Cell Transplantation, CAST, the CAST, at the Karolinska Hospital from 2012 to 2017. Marcus Maurer is now head of immunotherapy and immunosurgery at the Champalimaud Foundation and professor at the Medical Clinic of the University of Mainz in Germany in host-directed therapies. He has published more than 310 scientific articles, 28 of them in the Lancet series. He has 9 patents and serves as a reviewer for several international grant agencies and scientific journals. He's advisor to the Bill and Melinda Gates Foundation, advisor in infectious diseases and host-directed therapies to the UK Parliament, fellow of the Royal Chamber of Physicians in London. And more recently, unfortunately, for all of us, but fortunately, we had him as an advisor of the Italian government in the COVID vaccine trials. Maurer initiated with Sir Alimuddin Zumla, also recently, for the same reason, the Consortium Against Cancer and Infectious Diseases with a particular focus on COVID-19, hosted at the Champalimaud Foundation at the Center for the Unknown in Lisbon. Thank you, Marcus, for joining us today.
Thank you very much. Good morning, ladies and gentlemen. I'd like to share with you in the next 10 minutes one theme. Form follows function and location. This is not only applicable for smart, beautiful architecture. It's also, I would say, the title of biologically and clinically relevant academic alive research. And it would be even superb if beautiful and smart architecture, form follows function, and location goes together. That clinical academic medicine meets architecture, that the patient is in the center, goes back and forth, and I hope that we're able to share with you some of the visions, but also some of the realities, because some applications in clinical medicine can only be met if certain requirements are handed over, and some of them are essentially met in a very unique fashion here in the new Boton Champalimaud Center. The search for cure which is a dirty word because I mean, cure cancer is something that appears to be lunatic. It appears to be the mission to the Mars that Dr. Fior was just sharing with us. But essentially, we should not keep this out of our view. This is Professor Rosenberg. He was a very young fellow, over 30 years, appointed chief of surgery oncology, chief of surgery at the NIH. This was 1985. The search for cure, now we are here. And if we look at the most deadliest cancer, one is brain cancer. It's about 14 months after diagnosis. Actually, what we have been able to add in the last 20 years is exactly 2.6 months of survival in addition. A similar horrible disease is pancreatic cancer. Only 2 out of 10 individuals can be operated because patients are diagnosed in very late stage. And in 2030, we expected about its number 2 in the newly diagnosed different histologies of cancer. So this is one of the reasons why the Breton Champalimaud Cancer Center is there. And what are the weapons? There's radiation. It doesn't really work in pancreatic cancer except for metastasis, some sort of metastasis, chemotherapy, and immunotherapy. That's the big question. One precaution, immunotherapy is one pillar of a multidisciplinary approach. So it doesn't solve the question. Without a functioning immune system, there is no magic bullet, there is no magic cure. So the patient needs to have a functional immune system in order to combat cancer. But there is a success story, because otherwise it would be stupid to go to Mars or to the moon if there would not be light at the end of the tunnel, if there would not be an example that it works, at least in some diseases or in individual patients, for instance, in patients with pancreatic cancer. This was one of the first patients that Dr. Rosenberg treated in 1985. And one can fully rightly ask, this is a long time ago, why haven't we done better for other diseases? The first disease that was tackled was patients with melanoma. When I studied medicine, it was a deadly disease. This is not the case anymore. This is a historical image here of a lung with a lot of metastasis. 85, 2 years later, 87, the metastases are gone. This lady is still alive. And the revolution is the following, because if you look at truck companies, we sell things off the shelf, pills or infusions. And about 1.5 years ago, that's the first approved living drug. The immune cells of a patient that are taken out and educated and given back. It's a registered drug from the Food and Drug Administration that is being reimbursed. So in a way, medicine has seen a revolution, because there is not a drug that is defined by a chemical nature, but the behavior of the patient's own immune system. And there was, I would say, an explosion of companies and academic efforts in order to make it work. It does work for patients with melanoma. 60–70% respond if they did not receive any other immunotherapy. This drops down to 20 or 30% if a patient had already any form of immunotherapy in patients with melanoma. But what about epithelial cancer, colorectal cancer, pancreatic cancer? It looks disastrous. It's about 5%, maybe up to 15%. Why? Because these immune cells that were successfully grown from a tumor, and I will come back to that, first of all, are inherently difficult to get out of this cancer, because cancer is a very adverse environment. Immune cells don't thrive there. And if they are being expanded, if they are being grown, they may not be the smartest ones. They may not be the ones that are able to recognize cancer cells. And the team here at the Champalimaud Foundation has mastered both. So every patient that undergoes surgery, there were about more than 120 pancreatic cancer surgeries in the last 3 months. Most often, if not all of them, immune cells can be expanded, and we are aware that the patient's own tumor cells are being recognized. So we have a little fellow here. It's Bono. It meets 96% of the genes. Bono and ours are identical, just 4% of difference. But there are vast differences between this fellow and us. One might think so. Now, there's diversity, because what I'm hinting at is essentially what makes a cancer disappear? What is the immune system seeing? What is the decision making? And there are many ways, of course, and we heard this from my colleagues. What cancer is, it's chaos. It's a tumor microenvironment. There are many factors. There's one factor, at least one factor, is our genetic differences. And we are all different. There's diversity that's being celebrated. And this diversity is also present in cancer cells. So cancer cells are very much similar as compared to healthy cells. And if there is a biological knife, if there is an immune response, it must be able to differentiate what is a healthy cell and what is a cancer cell in order to avoid collateral damage. So this is DNA. So what you usually do is varies, like you have the green into 2 strands, Watson and Crick. So you have green and the purple and so on. And then sometimes there's a thing, it's called a mutation. It's just one mistake. And sometimes cancer starts with just one mistake and multiplies and chaos starts. And what we did learn in the last 20 years, that immune cells from patients are able to detect, like sniffing dogs, this one single mutation. This is what makes a cancer cell different, even if some cancer cells have 60, 70, 120, or even several hundreds of mutations, and different, even at different locations. Form follows function, and location, where are these mutations, are immune cells able to see them. This is what one mutation can do. It's a horrible disease. It's a young individual's age. It's just one mutation, essentially. So it has vast biological consequences. And this is what we see with cancer. It's location, because people usually die from metastasis, not from the primary cancer. It's the metastasis in the brain and metastasis in the lungs. So essentially, in order to combat metastasis, this is what, in part, is being focused at the new Champalimaud Center, is difficult to treat cancers. So only the primary cancer. It's the patients that come to us with metastasis, and this is where most individuals die from the brain metastasis in the lung or in the liver. Uncontrollable disease. So we are sharing about treatment options, treatment modalities for patients who have no other options left. But we did learn, otherwise we would be stupid to engage, that it is possible. And a big example are the patients with melanoma who were treated many, many years ago and are still alive and among us. There's a different there are different cancers between individual 1, 2, 3. And even if we go within one individual of a patient, there's a diversity of cancer. Each is marked with a different color. Now, before we come to the building, and there's the reason why I made this preludium here, is how do we get to the right immune cells? How are we ensuring that there is not a blind flight? How are we able to make sure that we take the right decision, that the patients get the right cells back, some sort of mini transplant? The cells are trained outside of the body and giving back to the patient. So there are 3 forms of immunotherapy. So these are more aggressive forms of the immunotherapy, and this possibility, one is the immune cells They know the enemy, they know these mutations that are in the cancer, but they lost the battle. So one possibility, and this is how the entire program started, is they're taken out, they're educated. There are smart cells and less smart cells. And the challenge is here, which has been mastered here at the institution, that only the smart cells, not the bystanders, not the onlookers, but the wise ones, the good ones that recognize the mutations, they are being expanded. This is possibility A. Possibility B, the battle is lost. The patient has no competent immune cells to recognize the cancer cells. The patient either never had these cells or they have been destroyed from the cancer cells themselves. And one way is essentially to have a blueprint of success from patients who were able to battle cancer successfully and to transfer this message by genetically engineering into the patient's own immune cells. So we endow the patient's own immune cells with weapons in order to recognize the cancer cells. And this could be done with different technologies. And a different one, and we don't go into this, that these competent, smart cells that recognize cancer cells are not in the cancer, they're hiding somewhere else, and they may be teased out. Each of these different instruments, like there are different chemotherapies, the different ways of radiation, there is not one form of active immunotherapy. So all these different forms are important really in order to offer a smart repertoire, individualized for each patient, and this will be possible at the new Champalimaud Cancer Foundation, but there are requirements that need to be met. There need to be so-called GMP, good manufacturing practice facilities. There are constraints in order to provide these kind of therapies. Now, something very practical, and then I come really why this building is important. That's a liver. This wonderful liver has been resected with a big tumor. You have different areas of the tumor. Right in the middle of it, the tumor is dead, it was killed by the immune system, and this is number 2, number 3, and 4, 5. So go far away. What you see here are different immune cells. But what is most important, KRAS reactivity. KRAS, as we call it, it's a cancer driver. It's something that went awfully wrong, and is a mutation in a gene that is not only important that the cancer cell is a cancer cell, but also this mutation can be recognized. And if we look, these cells that are recognizing this mutation and are able to kill off, to contain cancer cells, are only here and only here, but not here and here and here. There's a different tumor antigen. If T cells, if immune cells recognize that protein, they are found here and here. So where should we cut? Very basic question. And this is a histology. This is if we magnify that. Each of these dots here is an immune cell. So if we cut here, we have a lot of immune cells, and hopefully they're smart and we're able to make something good out of them. If we cut here, we'll be very disappointed because nothing will grow. These immune cells are not there. So where are these cells? They are different areas of the tumor. This is the center. This is usually the battlefield. There's not that much left. This is the rim, and you see these few cells here that are at the border of healthy cells and the tumor cells. And there's something very important that I'd like to draw your attention. This is this mutation of KRAS. Almost 98% of patients with pancreatic cancer have that mutation. And what we do see, and this is a hormone of the immune system, it's called gamma interferon. Gamma interferon works not only on the cell that is attacked, but also on the neighbor, and the neighbor, and the neighbor. There needs to be enough of this protein around. And this is only recognizing the mutation and not the wild type. So it's a biological knife. Where do we cut? This is part of the Champalimaud Cancer Center. This is the surgical suite here. The tumor is being taken out, but then there are few doors here. And they go directly to pathology. They go directly to the immunotherapy lab. So what we do right now, because we have to scrub in, we go to the surgical suite, we look at the tumor in order to make a decision not to make a blind flight, but essentially, where are the best cells? Where are the best cells in the cancer that are able to do the right job and that are worth to be amplified from a few hundred to a few billions, to trained outside the body and to be infused back. This is happening exactly in this room that is prepared. Form follows function, and we're able to locate them. So the pathologist is able to say, well, don't go there. What you see is all tumor cells, and the dark cells, that is our immune cells, go here. They're right there, and they're not there just by chance, because if they are very close to a tumor cell, we are able to catch them in the act. So this is not just a random, you are attacking your neighbor. If you are able to attack your neighbor, you may be a good cell to work with and to amplify it. You have your sister, your brother, and so on. So you go from 1 to 2 to 4 to 8 and make millions of these cells to treat. Here, for instance, we have such a specimen. This was removed from one of our excellent surgeons. More recently, here you have the artery, right? And the cells go out here. That's the necrotic area. This would be a blind flight. This is done in each center in Europe or in the US. And that's the unique situation here. So the pathologist says, well, I mean, the cells are here, or here, or here, or here. So we know exactly where to extract the smart cells. Now, what are we going to do with the cells? We're going to analyze them. We need to know them better. And this is part of the research facilities. And what you see behind here is the so-called GMP rooms. GMP is good manufacturing. It's a very dedicated area. You could imagine it's an operating theater with all the restrictions, because essentially you take the immune cells or part of the immune cells of the patients out to these very particularly dedicated rooms, and you need to keep them for 2 or 3 or even 4 weeks in order to nourish them, in order to train them, and then to be able to give them back. And this needs an investment, this needs structures, and this needs knowledge. And there are only a few centers in Europe which do have these, and the same is true for the US. It's a prerequisite. Otherwise, these therapies cannot be applied, or they are available, for instance, from big drug companies who have a center in the US, for instance, in New Jersey. The cells are taken out, they're shipped there and manipulated, and are taken back to the hospital. This is personalized therapy. The patient is in the center, and form follows function, and the location is really identified directly during surgery. Now, what we are able to do, or what is already happening in these research areas, is we're looking exactly at the enemy. We're looking at all the different genes that are mutated and all the different mutations that are going on that could be targets, that could be recognized by these cells. Again, all of these 3 possibilities to train the cells outside the body, or if the patients never had these cells, we're able to put, by using genetic engineering technologies, the knowledge back, or to retrieve the immune cells from a different area. So this is essentially, you're able to look in a GMP facility. It's not in the basement, it's transparent. It will have glass walls. So there will be there's a possibility to genetically engineer cells, to endow them. And this is not only future music, this has revolutionized some treatment cancer in patients with hematological cancers. Before, patients got a stem cell transplant. Now there are big companies who provide very expensive treatments, which are able to cure some of the cancers. Hematological cancers, cancers of the blood, This is not true yet for patients with solid cancer, for instance, pancreatic cancer. We have used different vector system, CRISP technology, also non-genetically engineered cells. There's a possibility to long-term storage the cells, because some people undergo surgery. They're okay for a few years. Would be good to have their cells frozen, ready to fight if the cancer comes back. And this is being prepared for. There's a functional monitoring 24/7. It needs certain requirement. It's a knowledge. It's the people. There are certain platforms, but also less aggressive, what we call tumor vaccination, that we stimulate and use the patient's own resources in order to do things. Now, how these are the cells expanded? There's a nice toy. It's called a cocoon. Maybe some of you saw the movie Aliens, or they're in cocoons and something comes out. We also have cocoons here. If these cocoons are open, essentially what you do see here, these are nourishment of cells, and then this is a structure that mimics a tumor microenvironment, and we focus in here. If we focus even deeper, we see these beautiful cells. Each cell here is one immune cell. These were cells that were extracted from a patient with pancreatic cancer about 3-4 weeks ago. So they can be extracted from a small piece, multiplied to billions, and trained outside. We need to know them much better. So each of these cells has a name tag, has an identity. We're able to color them. And each of these name tags is here. Essentially, each of these cells, we are able to have a molecular name tag. So we're able to trace them. There's a lot of interesting and clinically relevant basic research going on in order to learn from successes, but also from failures, and how are we able to trace these cells over time. No blind flight. Form follows function. We even want to know the function down to the molecular level. Now, here's the reality, what's happening. These are tumor cells that have been extracted from the patients here, and these little fellows that walk around here want to kill cancer cells, but this is the totally different story. That's the cancer cell that kills the tumor cells. This became a T-cell cemetery. This is why, in part, why tumors develop. Because they kill the immune system. They kill the small cells that try to attack them. There are several ways to leave your lover, Simon and Garfunkel. There are 50 ways of the immune cells to kill cancer cells. And we need to know them all in order to overcome them. For instance, here, this looks like a little bit moonlandish here, but that's a nice story. That's the cancer cell here. And there are immune cells. Here's one, here's one, here's one. Here there's this little fellow attacking. And then, see, this tumor cell just become flapping, just dead. Dead as a doornail. We can reverse this process. And this cocktail that is used in order to do that is different for patients with pancreatic cancer as compared to what is used for patients with melanoma, because it's a different microenvironment. And this team here at the foundation was able to identify this cocktail, to make them smarter, to make them survive in order to do exactly that job, namely, not only a few cells, but an entire soldier, an entire army in order to kill cancer cells. Now, this needs to go back into the patient. This needs to go back, and we need to have the cells, the T cells. We can do that. The prerequisites are here. So we're able to produce these cells to the back. Of course, it cannot be infused because we need to have the building, we need to have the structures, and we need to have the proper guidance of scientific committees, of safety committees, and from regulatory authorities. So it's a thing of political will. It's a thing of structure. Cytokines, these are hormones of the immune system that if we infuse these poor cells that are able to survive and are really in a good spirit to do the job and to be long-lived. Safety. Something could go wrong. Collateral damage. So we need to be able to control that. And this happens in intensive care units that are being prepared for, also at the foundation. So it's a team, not only, it starts with the patient, it goes back to the patient in order really to follow these cells, and if there is any collateral damage, that we're able to go in immediately and stop if collateral damage may occur. So to summarize, it may, before we said we take a biopsy, we take the tumor out, it may start in surgery. Maybe the future is even before. Maybe we're able to visualize where the immune cells are, the smart immune cells are, the good immune cells are, before we operate. Do we take this metastasis or that metastasis? So imaging and tracing of smart cells is an important challenge. This goes into the surgery. No blind flight. Form follows function. So the T cells are being, or areas of the tumors are being identified and recognized in a sterile environment directly there where the surgery takes place. This doesn't exist anywhere in Europe. A similar facility may be built in Shanghai. So it's a novelty, but essentially it brings something together that a lot of people are aware of. But here it is being exercised. The GMP facility, that's the facility that is needed to keep the immune system alive or to genetically engineer. It's a big investment. It's a structure that needs certain functions and supervision, constant supervision by the regulatory bodies. And also to be able to go back to give these cells and to trace them. So to finish, form follows function. Certainly, it has already been materialized in a building that you are going to see existing and will be soon inaugurated. It's the start to allow serendipity in academic medicine that becomes a reality and is able to materialize in the building, but also materialize in better treatment, diagnosis, and care for patients with difficult-to-treat cancers. Thank you very much.
Thank you very much, Marcus, for this wonderful introduction to this new endeavor at the Foundation. We are running a little bit short on time, so we won't have much time for the discussion, but we would be able to take a few questions from the audience. And I would ask you also if you could just take the blackout a little bit out while we have the discussion before fully opening the window. Any questions from the audience? We have microphones going up and down. We have a question here and a question there. The lady here in front first, I think. No? Okay. Please. Go ahead.
Okay.
You speak
okay, I can go ahead.
Yes.
If you can hear me. And first of all, I'm Martin Kokk, I'm the ambassador of Estonia. And I would like to thank you for that most interesting presentations and also for all the work you've been doing here for the good of mankind. But the question I would like to ask is that there has been a tremendous progress in treatment of cancer in the last 40 years. But this progress has still been a bit unequal. There are some types which get better treatment, some types of cancer which are more difficult to treat. And my question to you is that can you make this sort of generalization? What has been the obstacle in some cases? Is it a regulatory environment? Is it some sort of financing issue? Or is it just poor luck that in some cases the breakthroughs are coming through and then the others we have to wait a bit longer? So thank you very much.
Marcus, would you like to? We have all taken the shots and we are tested on a weekly basis here. So while we speak, you're welcome to take your mask out and not speak to Rita.
Thank you very much for your question, which is which is a very important one. I would say the headline is democratizing access to academic medicine. And there is an inequality between different countries and different areas, and access, and there are different ways to overcome that. For instance, the treatment of patients with hematological cancers, how it was solved. Essentially, this was solved that there are big companies in the US and now also in Europe, and it was a question, how can we solve this? And this was solved by to make a few centers. For instance, for one center is in there are two centers in the US. One of them is in New Jersey. Another one is in Amsterdam, here in Europe. So the cells are being shipped to the center, and then the patients are being treated. But this does not solve, doesn't answer the question regarding the personalized medicine. One question was essentially, is it regulatory? I don't think so. I think our colleagues at the regulatory bodies are very smart, very helpful, and very insightful. I think it is really the drive of academic medicine to make a decision what is the political will and the investment of offering personalized medicine. For instance, if we look for a totally different example, stem cell transplantation. It is solved in Europe in a way, more or less. If you look for a patient with a rare hematological disease, you go to a center. You go to a center in Estonia, in Russia, in Germany, here and there. And there may be potentially centers of excellence that provide these more private, excellent treatments for difficult to treat cancers. And I would be able to say this democratizing access to academic medicine, the model of how hematological allogeneic stem cell transplantation is handled may be very similar for these difficult to treat solid tumors. This needs a political will. There is certainly an absolute support from the regulatory authorities, but there needs also being investment that one treatment, for instance, if we take an off-the-shelf treatment for the FDA-approved treatment for melanoma, it's a whopping $380,000 treatment. This can be different. So one possibility is to have insurances, state insurances, and private insurances very early involved. But this is a question to you, to the policymakers and the politicians. I think because it's always funny when people say, oh, this is so much expensive, we're not able to afford it. But I think it is affordable if people are involved early on. And maybe some academic centers in Europe, and also like here, are able to exercise that things can be done at a reasonable cost and effectiveness. I think there is a very good way in order to do that. I hope that this, in part, answered your question.
Thank you, Marcus. There was a lady here on the front row, and I'm afraid that will have to be the last question.
Thank you very much for this impressive presentations. I'm from Slovenia, Corporate One Ambassador from Slovenia. I have basically more a question for the last speaker, but maybe also for the others, and namely about the relation between genetics and developing cancer. If I'm informed correctly, it has been proven in the research that genetics plays a key role in developing breast cancer and maybe some other cancers as well. But do you have any other information about other types of cancer which are clearly where genetics plays a key role? And linked to this, is there anything you would suggest as a preventive measures to anyone who has a family history in developing cancer? Because I've heard here primarily that you focus on treatment. But are there any other tests or anything else that can be done as the preventive measures? Thank you.
Everyone gets to the genetic comment. Maybe Enrique and Rita would like to comment on the genetics and development and the prevention and the medical advice. Maybe you could take over that, Marcus.
Okay. So, I don't know if you hear me. I don't think it's
It's
working.
You
Okay.
have to be very, very to the point.
Okay, so related to prevention, I think it's screening, that now people are doing more screening. So if you have a family history, you should do the checkups related to the type of tumor and the risks that you have. And so I don't know, what was the other question, maybe?
Genetics and cancer.
Yeah, genetics. When you have this, well, you can sequence and know the genetics, but basically then you can do the prevention by screening, or like Angelina Jolie. I think it's the example that she just you can take the organs out as a prevention, but mostly I think these extreme cases where you do screening or you try to get rid of the problem before it comes. But maybe I'm being too simplistic.
Enrique, would you like to comment on genetics and cancer, and Marcus?
Am I on?
You're on. You just need to start speaking and you're fine.
And in cancer development play a major role. They act as I would call it a seeding effect. But if you've got a seed, the plant only grows if you find fertile soil for that. And that's where your behavior in the environment makes a lot of differences. So a mutation per se, and we all have cells here. Everybody sitting in this room will have mutations. They can come either by being germline encoded, so you were born with it, or they develop throughout your life. That's normal. The problem arises when you accumulate different types of mutations, so when your genetics go wrong. So you need a fertile soil for the tumor to grow. So in addition to the genetics, there's a very important component of how you behave. What are the environmental factors that can trigger that? And at the moment, the best weapon we do have is, as Rita was saying, regular screening is prevention medicine.
I think there's nothing to add from my side. But probably you want to hear something very practical. The practical advice is, of course, the screening programs. This is what in the EU is being the genetic tests. There is imaging, but there may be one point that is still out there, and there is some shortcoming, namely active prevention. Because what you do is you're waiting for something in order to catch it early enough. But I think there's knowledge out there. With smart medicine, and we do know this. One buzzword is the microbiome. So we know that we are not living alone here. We have a lot of bacteriophages, viruses, bacteria that induce inflammation or stop inflammation, good and bad guys. There may be even preventive measures if you know your risk. We may be able to influence in a proactive way that development. The fact that there is a gene doesn't mean, essentially, the fact that you have a piano doesn't mean you're playing really the melody. And you're able, and if there may be smart ways in order to prevent that this melody of cancer is ever played. And I think this would be one proactive way, not only to early diagnose, but to actively prevent.
So there's several questions, and Ambassador Lurti gave us the possibility of having them answered. Can we have the microphone down here, please? I should also just add to the comment Marcus made at the end of his first intervention. This is an issue for all of us. Today we can cure many more cancers than we used to. And the incidence of cancer is pretty similar. However, we have many, many more cancers coming up. The number of cases are increasing every year. The same in terms of cancer deaths. Today, we die if we get cancer. We're much less likely to die of cancer, thanks to people like those on this stage, than if we got cancer 10 years ago. But the number of cancer deaths is steadily increasing because of the aging of the population. And that's a problem that we have to solve. There's another problem as large, and in terms of cost, arguably larger, which is dementia and the increase, the fast pace of dementias in the world and the projections of the numbers of dementia, with the costs not just being imputed to the health systems, but also to the indirect and social costs of treating people with dementia. And that's certainly a problem we all need to solve as a society. Thank you.
Dr. Evelyn, thank you very much. And I just do not want to overstay our welcome, but you are the ones allowing the questions to be answered. And perhaps if we can take a bunch of just 2 or 3 questions, we would really appreciate not to also overstay our welcome. I do have a question with a bit of context, as you know, and the people around here in this auditorium negotiated it. The new framework program for science, Horizon Europe, includes a new instrument, missions. One of the missions is the cancer mission. And the idea of its creation was very much the moonshot idea. What can we do to cure cancer? The dirty word that Professor Meyer talked about. My question is this sort of instrument helpful? Is this sort of instrument the sort of things that scientists were, from the public policy part, looking for a need in order to indeed, I guess, to cure cancer is a path, not only an objective, but is this helpful, or should it have been drawn in another way? Or what do you need from public policy actors? Thank you very much.
Maybe we could have Enrique and Joe comment on the funding models of getting from science and from deep science questions to actual treatments. What might work best, and whether these new mission models and moonshots and financing things under one outcome are
May I
helpful.
start by
Please.
tackling that? Well, there's no doubt these mission programs are very important. They are dedicated to specific topics, and that really allows for the knowledge that is out there to actually get together, frequently in consortia, that can tackle really big questions. Science and medical practice in general and big developments do require multidisciplinary approaches. But you think about really disruptive knowledge that can completely change the way we're looking into cancer specifically in the next 20 years. That really comes from very basic science. Just to give you an idea that we all went through it. The reason why we actually make vaccines in 1 year for COVID was because there was a fundamental investment in basic research. And curiously enough, the most successful ones come from scientists based in Europe, Moderna and BioNTech. And that's a fundamental step change that we need to do in European science policy. Not only strong and bold investment on basic science, but very important, what is missing is the bridge between the knowledge and groundbreaking innovation that we're doing in our laboratories into products. And that's where the US makes a huge difference to Europe. You have fundamental discoveries in the US that, as a seed funding from venture capital, can raise anything up to 100 million euros based on a concept, dollars. In Europe, we still have a very little sort of, I would call it, provincial mindset. We go step by step, 1 million here, 2 million there, 5 million there, and there's no bold investment on this innovation that can make really a big impact. And that's the reason where the science behind Moderna actually went to the US, to the Boston area, and didn't stay in Europe. So at this stage, we could have BioNTech and Moderna based in Europe having a huge impact in fighting these epidemics. And actually, everything is being developed in the US, based, in essence, in expertise that was born and raised in Europe. So I think this needs to be really complementary, with a very strong investment in science, in translating basic science into products, and then indeed these very important and big mission programs that allow people such as Marcos and other colleagues to bring that knowledge into practice, into the clinic, into solutions for society.
Joe, would you like to comment?
Sure. Cancer is not my field, but in terms of the history of science and medicine in general, I think we have to be mindful of it, because what history tells us is that the knowledge that's generated from fundamental research is fundamentally necessary for developing applications, but often in ways that are completely unforeseeable, right? So I think in terms of these big investments, one needs to have a kind of balanced portfolio. One needs to understand that investment in fundamental research is often a very long-term investment, right? Now, we've heard some cases where you can go very quickly from basic research to applications, but the vast majority of times, the impact of fundamental research is seen 10, 20, 30, or even longer after the initial work is done. But what we're seeing in a lot of different places is that there's an increase in funding of so-called translational or applied research, and there's a steady decrease in funding for basic research. And when the NIH detected this several years ago, they freaked out, because they know that the source of all the ideas, the source of all the knowledge that drives the development of applications is ultimately fundamental research. So I think, I'm not saying you should invest in that at the expense of trying to develop applications and translate that knowledge into applied solutions for health or any other area. But if you take it away, the well will dry up. So, I mean, I can talk about cases in neuroscience. So take Parkinson's disease, okay? The frontline treatment for Parkinson's disease today is dopamine replacement therapy, okay? That was discovered because researchers in the 40s were trying to understand if chemical neurotransmission was present in the brain. They weren't trying to study Parkinson's disease. So they started depleting certain neurotransmitters, or certain chemicals they thought might be neurotransmitters. And lo and behold, they observed that depleting some of them produced catatonia in rabbits. They then put in various precursors to various chemicals and found that the dopamine precursor, L-DOPA, rescued that catatonia. At the same time, in Austria, there were physicians and researchers who saw those results and began to look at postmortem brains of human patients who suffered from Parkinson's disease. And they found that it was the cells that produce dopamine that were dying. Within a very short amount of time, L-DOPA, dopamine replacement therapy, entered into the clinic, okay? And that was driven by literally no focus on a disease. So I really think in terms of setting policy, you need to have this balanced portfolio, this balanced investment in basic and applied and translational research.
Thank you, Joe. Thank you, Enrique, for your... We have another question here, and...
Thank you very
question?
much.
Okay, perfect.
Thank you very much. I'm coming from Germany. So I have two questions. The one was already now touched upon with the vaccination issue, because what we learned in Germany was as well that more or less the finding of the vaccine was a byproduct of some kind of research on cancer. So how is then the prospect going for the cancer curement, let's say? Because of course now the focus is on the vaccines, but I understand that something more profound is behind it concerning cancer. So do you think then this what they were finding there will be a kind of curement for not so long from now? Is there something really that is groundbreaking? First question, and second is about the democratization of this kind of research, what you're doing here, and curement of cancer, because what I see is obviously a very expensive way of here treating, and this is for special patients. So I understand if we want really to be successful in that, we have to transform this to, to the world around, let's say now in Europe, to make everything what you are doing here, also to apply this to smaller clinics and to really to cure a lot of people. So this kind of transformation from what you're finding here, how this will look like then to go to the other hospitals. Because in your clinic here, I understand very few patients can be treated, and I'm sure that no German insurer can pay the cost for that. So I think we have to do something about that to transform it to a kind of broad application if we want to be really successful, and not just to be in a kind of here small tower where we are treating some patients who are lucky enough to get it paid. Thank you.
Thank you for your question. Before I pass it on, I just have to make a comment. It may have you may have had the impression that at the foundation we only really pay patients that can afford a very high cost of medicine, and that is not the case. We see patients from many health systems, including many of the systems of the National Health Service and the usual insurers. And in fact, we even see patients with German insurance. So, so I just wanted to make that comment. The foundation was designed, and I even had a little presentation, but I didn't want to take too much time. But the foundation was designed with patient-centered care in mind, and patient-centered care in mind across the whole pathway, not just in terms of the access to the medication, but there are many beautiful things you can see if you go into the path, into the foundation. Small innovations in technology, from the fact that patients get a smartphone and they're never called their name loudly and can occupy any of the space. The fact that a patient in a room that is here, when the bed rises, the table moves with the bed and he can just take his glass and does not have to call the nurse in order just to drink his water. And small things like that you will find in robotics, in many small innovations that are really driven by the wish of the foundation board, the scientists, the engineers, and the physicians here working together to make that patient-centered care not just a word, but something that is real and accessible. Sorry, I just needed to say that. And to comment on the first question, also you've already hinted a little bit at this. Maybe Marcus, you would like to continue.
Be delighted, because in a way Mainz is my the stomping ground. If you look at the history, it's a huge success, because it's a conglomerate of 7 companies: Ganymed, BioNTech, and one of them, Drone. Drone is a non-profit foundation also that works with the university together. We're looking here at the result of a 30-year investment. So it was a 30-year research and serendipity, a few publications, a creation of intellectual property, and an immense amount of building of structure and expertise. So essentially what the byproduct of the RNA vaccine was essentially starting small cancer trials with cancer vaccines. Started with Drone and BioNTech in terms of, so that's the success story. This is exactly what Moderna did in Boston. It started as a cancer vaccine study. So essentially this interface between companies, non-profit organization, and it is a success story. It tells us also it took serendipity in 30 years in order to achieve that. Second point, very quickly, why is BioNTech and Moderna, what is the treasure? And I think here is the European idea that we could do better, because essentially there are, no matter if immunotherapy, yes or no, or chemotherapy, the long-term survivors, short-term survivors, and so on, each center, each university has its own sequencing facility. I think the dream would be, and that's an amazing starting point, everybody who has cancer and enters not only here, but in Mainz or in Estonia or everywhere, should have their cancer sequenced. It's not done. It's not even reimbursed by most of the health insurances. They sequence 160 genes. So this would be something that is achievable. It's a policy point. And your last point was essentially, how can we get out, is this an elite structure? It is not. This is what you already heard. But I think what we need to learn is essentially what works, not what works. Whatever works is affordable. When people looked at artificial kidneys, it will never work. It's much too expensive. Stem cell transplantation will never work. It's too expensive. If something works, like a tumor vaccine, it will be made affordable. If a T-cell therapy works, people will find, and this has to be found out, are there particular centers? Do we refer patients to a different center? Are there centers of excellences? So I think it's the serendipity with the knowledge of the Modernas and the BioNTechs, with the will to increase knowledge and apply warehouses of information that does not exist in Europe. Some universities do it. Sequence each patient's tumor, they deserve it. This knowledge is needed. And to identify small structures in which it works and essentially see what works for each patient and works for each region tailored for each country. There will not be a perfect solution, but I think this is this could be a way in order to make it workable.
Thank you, Marcus. We have one, it has to be the final question.
Thank you very much. My name is Maeve Collins. I'm the Irish Deputy Permanent Representative, and I found all of your talks really very, very interesting. I have two questions. The first, maybe the neuroscientist, but maybe all of you could answer, is really on disinformation, because for all the progress that science makes, science has many enemies, it seems, at present, with very powerful tools at their disposal. And what can policymakers and researchers like you do to counter particularly vaccine disinformation, but also distrust of the health system and distrust of science? My second question was, I think, rather similar to Susanna's, which was really, how do you bring what you do to the model that I am more used to, which is the public teaching and researching hospital? I would just like to know a little bit more about how do you get the progress out of the boxes here and into the wider system, and how can it be done more quickly? Thank you.
Joe, would you like to comment on at least the first and the second, and then maybe Enrique as well, as both of you head Champalimaud research and have responsibilities in these areas?
So, I mean, this doesn't so much touch on my expertise in neuroscience, but I think, you know, one of the answers to your question is that we need a lot of investment in education at all levels, right? In the US, you know, there's been a sort of steady decrease in public education, which I think makes fertile ground for disinformation. Also, as time goes on, you know, the way we're sharing information is changing rapidly, right? And networks, basically, closed networks are being created by things like social media. I'm often amazed when I see the outcome of, say, elections with respect to some of these issues, because I've never met anyone who holds the opinions that are driving the voting of a lot of people. You know, we live in these very, very separate bubbles, and I think that is a potential breeding ground for a lot of bias, right? And so I think it's a very hard problem. I don't pretend to have the answer to it, but I think we have to find ways to bring people back to the table of a common reality, you know, that's based on reason, logic, evidence, right? And less susceptible to our own kind of inborn social biases. And you know, the kinds of ways that we teach people to think within our own educational programs, I think, are focused on trying to help people think critically. How do you evaluate evidence? But that needs to happen, you know, on a much broader scale, I think, if we're going to push back against disinformation.
Enrique, would you like to comment? I would just add, even though you won't have much time, Rita, I was just this week in a school in a very difficult area in Lisbon called Cova da Moura, at the invitation of Rita, who was taking some of her time, her lunchtime, in fact, to just be in a high school, probably in one of the most complicated areas in the Lisbon metropolitan zone, and going back and forth and doing this is important.
I fully subscribe what my colleague Joe just said, but I think the problem is certainly as a scientist or as a scientific community, we can contribute to fight disinformation. But I think the vast problem is much broader, and it has to do with a broader education system and social inequalities that we live in. And I think it would be worth it to listen to what Rita might tell us about that experience on this education program that was established with poor neighborhoods in the big Lisbon area, exactly to tackle some of those problems.
Okay. Hello? Yes, this example by Andrea. So Andrea joined me this week. It was this week that we went to the school. And yeah, but the idea is really to take, so we have, I think we have a problem in Portugal. Maybe this problem is across Europe, which is in our country. We don't see this people that from poor neighborhoods going into university. So I give classes also in university. I guess all of us do that. And then you don't get these students coming from these difficult neighborhoods going into higher education when it's free. So it's free, so it's not a problem of that. They're not getting access. So this year we started here at the foundation this project to try to start building some connections here in these difficult neighborhoods to bring these people, to take them to higher education. The other example, I guess, is tackling this process here, that this problem of, I think, this trust in science and whatever. It's really disturbing, I think, how people get into this misinformation. Is that here at the foundation also other scientists here that started, Marta Moite, another neuroscientist, and we were all together, a lot of people here from the foundation, but then all the research institutes, first in Lisbon and whole Portugal, that we teamed together to go to schools to have, well, we didn't go, we did it by Zoom, but to have sessions to explain about the vaccine and to, it's open questions, if the students have questions, if the teachers have questions, so try to educate the children in all the schools in whole Portugal. And we did a campaign, and we were involved, our institute, and in fact Marta was the one, the driving force, and then from our foundation, and then the whole country was involved in this. Maybe we should do more, I think, probably, but we are trying to be at least attentive to these problems and trying to act. Whether it's going to be the right answer or not, but I guess we should try all as a society to try to demystify these problems. And well, the vaccine is actually a very good example how some people are still with crazy ideas about the vaccine. I can only say that I was very happy to get my shot this week, and I think I don't know what more to say.
Make a quick comment.
Follow up on what I started saying about social media. So in AI, there's this term called the alignment problem. Okay, it actually comes from economics, from situations where, for instance, the incentives you create in an economic system can end up having unforeseen and undesirable consequences, like increase in the disparity of wealth within the population. But applied to AI, these are cases where you try and set up an algorithm under certain objectives, okay, which may have a positive, outcome, or you want to have a positive outcome, and they end up creating these unforeseen negative consequences. So in the case of social media, for example, the model is, let's design an algorithm that maximizes this objective function. Amount of time I put something that you're looking at our platform, right? And then the algorithm's just going to go from there, and it's going to start learning, and it's going to start putting things in front of you that maximize the amount of time that you look at it. And what happens if you multiply that effect out over long amounts of time and over enough people is you get these bubbles, right? And you get these alternate realities. So I think from a policy level, people should be thinking about how do you regulate these algorithms, okay? They're incredibly powerful. They can bring a lot of benefits to humanity, but there are also a lot of risks, right? So, yeah.
We will have to close now. These are important issues that we all have to deal with. The foundation is an ideal place to get this multi-stakeholder teams thinking about these things and acting in the world, involving students, involving education, and also involving patients. We have this week a forum with patient organizations in Portugal and then across Europe. And one of these things that the new funding mechanisms also allow is to listen to the patients. Listen. No one knows more about the disease than the patients. Listen to their experience. Work with them to even formulate and prioritize research questions. From basic research, which is, as you heard, is extremely important, to its applications. And that's another thing. It was a real pleasure to be here. We really enjoyed this common discussion on what I see are our common concerns and goals. I apologize for the delay, and we will be coming up for drinks. I believe we have to take a picture. Will we be able to take it here or outside? So we will go out. We will be walking out, and we will be walking in the direction of the outdoor amphitheater, which you saw in the initial movie. Thank you so much for your time and for being here with us.